My name is Ryan Brown. I am a clinical scientist. I specialize in clinical chemistry and clinical immunology. So the areas of the blood and body fluids that I like are the non-cellular components, okay? And that's what we split it up usually, the cellular components and those non-cellular components. And I just want to thank, Eric, is it? Eric, thank you for giving extra credit. This is a nice turnout. Probably a little better, so I'll take it. Yeah. So anyways, so what I'd like to talk about today is I'm going to give you a brief description of what we do in medical laboratory sciences, but also give you information regarding really how cool it is, right? And how innovative this field is and how inventive it is. That's really what we see here, is the collision of science, right, true science, and the most up-to-date technology. So when we go through school, we don't really learn a lot of the technology portion. Most of that's done when we get on the job and our machines that we rely upon start to break down. So that's when we actually get to start using them. So that being said, what drives innovation for the laboratory? Why are we going to continue to create new things and innovate and become more efficient for the future? Well, there's a couple different things that are making the lab go into the future, or being forced into the future. Let's put it that way, okay? Because there's a lot of external circumstances that exist out there that are not under our control. First being population, right? We're having increased population every year and it's exponential. Right now there's 7.4 billion people in the world, okay? In 2050, there's gonna be 9.4 billion people in the world, right? 2 more billion people in just a little over 30 years. And if we continue the way we're going, right, where there isn't a total collapse of our system and we revert back to chaos and anarchy, if we continue in some decent fashion, which is sort of questionable these days to some of us, if we continue going the way we're going, we're going to continue to have more people in the world, and these people are going to live prosperous lives. They're going to live longer lives, but if they're anything like us Westerners, they're not going to have necessarily healthier lives, okay? So they live longer, but those last 15 or 20 years are a little rough for most people, okay, especially in the United States. So what do we do to take care of all these people? Well, we have to speed up what's going on, okay, especially in the laboratory. So just reiterating what we die of, okay? We're not dying of random things. Eagles are not flying out of the sky and scooping our eyeballs out of our head and dying of that, okay? We're dying of things like heart attacks, right? Myocardial infarctions. We're dying of stroke. We're dying of diabetes. We're dying of things that we can control, okay? That's what we're dying of. And for the most part, the laboratory, we take care of all these things, okay? We take care of all these things. Nephritis, okay? Kidney function, infectious disease, diabetes, obviously, we talked about that. Alzheimer's, I mean, look at how many people Alzheimer's kills, almost 100,000 people a year. And we know very, very little about this disease, okay? So the more people we add to this little thing we call planet Earth, the more these numbers are gonna increase, the more people that are gonna be in hospitals, okay? And the more work we're gonna have to do in the laboratory. So what's another driver for innovation in the lab? What's gonna cause us to keep creating interesting technologies and interesting methods of doing things? Well, the market is one of those forces, right? We all learn about Adam Smith's theory of the invisible hand in Econ 101, and we know that if there is a market for healthcare, people will buy it. And you can guarantee there is a market for it, okay? Healthcare spending in the United States accounts right now, excuse me, actually, this is a 2015 estimate, accounts for 17.2% of our gross domestic product, which means 17.2% of everything consumed in the United States is relegated to just healthcare, okay? In 3 years, in 2020, less than 3 years, we're gonna jump up to 20% of our GDP, okay? So 20% of all consumption in the United States is towards healthcare. So I ask you, is there a market, right? There is a market. So long as we have a market where people are consuming our goods, there will be innovation, okay? Because we need to keep up with market demand. So what's the last driver of laboratory innovation? The main one for us in the laboratory is caretakers, okay? Caretakers. Doctors and nurses want their results now, okay? They wanted them yesterday. They wanted them the day before yesterday. The idea being is we can't get it fast enough to them, right? And that makes sense because we want to help our patients as best we can. We want to prevent them from having any more cerebral damage from a stroke. We want to prevent further cardiac damage, right, from a myocardial infarction. So if we can treat them, right, in the beginning stages of disease, we have a better chance of healing them and they can live a prosperous, normal life. But that doesn't necessarily happen, right? And we've all been guilty of this. We feel sick and we wait way too long to go in the hospital to see a doctor, okay? And then this is what happens. We have this sort of collateral effect where everyone's waiting and next thing you know, it becomes a crisis and we go to the emergency room instead, okay? So you wonder why, if any of you worked in healthcare before, why nurses are stressed out, why doctors are stressed out, because they get to experience people in their worst condition, right? And not only that, one after one after one. There's no break in it. It's people at their most horrible, and you just have to deal with it, okay? So to take care of these people, doctors, right, have to have their results right now. And they have to be reliable results, and they have to be easily interpreted, okay? There has to be no gray zone. As of right now, even though it seems like we know a lot about the human body, we know actually very little, okay? We know very, very little. And doctors have this incredible job of knowing all the different tests that exist out there to help them aid in diagnosis in combination with clinical manifestations of that patient. And then they do their best darn job of figuring out what's going on, okay? So this brings me to the point that these doctors, even though they demand a lot of us in the laboratory, they are completely and utterly dependent upon us in the laboratory. 70% of all decisions, medical decisions, not all decisions, are based off the results you get in the laboratory, okay? I would probably gather to say that there's even more than that, right? I'm pushing more to 75, 80, depending upon the kind of questions I get when I'm working in a hospital. But the idea is they depend upon us to make diagnosis and to make proper prognosis and treatment options for those patients. Without us, they're shooting in the dark, okay? So innovation, okay? What are the factors that exist in the laboratory that would want to drive innovation? Well, I want to break down the testing process for you, okay? And we have a couple different ways we can go at making the laboratory more efficient, more cost-effective, okay? And more helpful to the caretakers who are treating the patients. We look at it in a pre-analytical way, right? Before the sample is tested. We look at it in an analytical way, right? When we're actually testing the specimen matrix. Then we look at it post-analytically, right? After we have that data. How do we look at the data as a whole, right? And all these aspects of the laboratory are all things that are being worked on right now to improve efficiency, to improve cost measures, and to improve patient care most importantly. So, what are we doing? Well, this is a drone in Zimbabwe that's delivering blood to people in rural areas. And we're doing this right now, right? We have beta testing programs going on to where if you live in a rural area and you either need to get blood to a laboratory or get blood in you because you're bleeding out, right? They'll just send a drone up instead of flying a helicopter, which is what they do right now, okay? It costs about $2,000 a trip. And they'll just send a drone out, drops the specimen off, comes back. There is very little time, right, that takes up for that specimen to get from one place to the next. So this is a pre-analytical variable. If we can speed up the actual time it takes for us to get that specimen to the laboratory, we can test it faster, right? Makes pretty decent sense, okay? So another part of the pre-analytical spectrum is getting the specimen on the analyzer and getting it tested, but not the actual testing itself yet, okay? Right up into the point. And we've had automation, right? Here's a nice chemistry automation line in a hospital. We've had these for years, okay? Beckman Coulter has been beta testing these 20 years ago. So automation, we thought for a long time was going to kill jobs in the laboratory. So you're going to take my job away from me. This didn't happen whatsoever. All it did was make our jobs more interesting. Some people would say just more difficult, but I like to be a little more optimistic. It's interesting, okay? Because instead of just science, we're learning about robotics and fluidics, right? Electrical engineering. So we go into a whole different field from what I was traditionally, right, used to learning, which was just pure science, okay? So now I'm learning about X, Y, and Z axis robot arms and all sorts of cool stuff like that. Again, I had no idea about this stuff before. So if we can get these specimens to the technologist to test them more quickly, okay, we can get a result faster and we can treat the patient faster. Automation does not take jobs away. It just speeds up the process for us. But automation is only one part of the process. It's taking the specimen and it's delivering it to the technologist or the analyzer. That's it, okay? So the next step, of course, is robots, right? Any next future step has to be robots in my science fiction mind, personally, okay? So what we have now is, and check this out, I'll give you a little demonstration. They set this up on a circular grid, right? And they set this XYZ robot arm up, and it basically goes to the different stations, right? If it's picking up a pipette tip, if it's picking up a reagent from the refrigerator, if it's picking up a specimen from the counter, delivering it to the analyzer, programming the analyzer and making sure it's doing what it's supposed to do, takes the sample off, puts it away for you, and all the technologist is doing is looking at those results, right? Because that's what we're trained for, that's the whole reason we're there, is to analyze the results that come off of these machines, and to also troubleshoot the machines when they go down, okay? So will this, honestly, very cool-looking robot replace me? I don't know. I'd be sort of okay to hang out with it at least. But I don't think it's going to replace us. All it's going to do is make our jobs, like I said, more interesting. You're going to have to broaden your knowledge base. So instead of just being science, you're going to have to look at engineering too, okay? So that's why STEM goes together so well, right? Is all these things work in parallel with each other. You can't just be a science nerd these days. You got to get into engineering, okay? You got to get into mathematics. Because we're all a part of the same nerd herd, right? As Deb Disco would say. Okay, so these are the drivers, right? The pre-analytical, the analytical, and the post-analytical driver. So what are some examples of this? So what we've been doing for years, and you've always heard it, and you've probably done it in your labs too, is you use test tubes, right? Use test tubes to combine our reagents or chemicals and our sample matrix to find out biochemically what the heck's going on. The only problem is we're using quite a bit of material in our test tubes, okay? These were designed to appeal to us human beings who need this big visual cue to see what the heck's going on, right? But machines have taken our ability to visibly see things away from us. It's taken that job away. Because this test tube requires a lot of material in it, not just patient material, but the reagents. And that's where the kicker is at, the reagents, the chemicals that we use in the laboratory to determine your blood glucose value, your liver enzymes, your kidney function tests. All these require chemicals. That's the main cost of the laboratory, okay? Not the machines themselves, not the technology, but the usable reagents, the consumable reagents. That's where the money's at. So what do? So we evolve. We go from one test tube, 3x75, has about a 5 mil capacity on it, and we go down to a 96 well plate. This is our next evolutionary step. A 96 well plate has a capacity of about 300 microliters, okay? So the next step was a 386 well plate that has a 3 microliter capacity to it. And now recently, we've switched over to a 1,536 well plate, okay? From one test tube to 1,536 in one fell swoop. That's efficiency right there. And that's the kind of ideas that come from the laboratory because we're constantly trying to think of how we can improve processes, cut our bottom line, and improve patient care, okay? So what's interesting about this, though, is we run into some variables that we hadn't dealt with in the past. So here's something for you that I didn't learn until I started getting into lab technology, right? The discipline of fluidics, how fluid acts, right, under certain conditions. So for a very long time, using larger macro amounts of fluid, like we just described here, 300 microliters, We can physically agitate these things to shake. Now check this out. These very small ones, the one that has 1,536 wells on it, it has a fluid capacity of 2 to 4 nanoliters, okay? It's super, super small. So check this out. So when we get down to that amount of fluid, we cannot rely upon simple agitation or even ultrasonics to mix that specimen, okay? We can't depend on diffusion either to mix the specimen. So this area where we exist in the laboratory, this 2 to 4 nanoliters, right, is in this, it's got the worst of both worlds when it comes to mixing, okay? So there is a major effort right now just to find a more efficient ultrasonic mixer, okay? So I find that pretty interesting. Okay, so that's the sample matrix. One of the biggest things we like to do in the laboratory is give the responsibility of testing to somebody else, okay? We need to get some stuff off of our plate. And the best way we do this is by giving the caretakers, right, who are directly responsible for the patients, some responsibility for testing those patients. And the way we do that is through point-of-care analyzers. We've all seen a relative stick their finger for a blood glucose, right? We've all seen that happen, okay? But now they're able to do something like take a picture of a wound, run that picture through certain filters, okay? Those filters can help us determine at least a semi-quantitative amount of microbiology or biological cultures in that wound. And so we can treat it faster. Literally just by using a camera and passing it through filters. What started this technology was the smartphone that I don't have now. Oh, here it is, okay? Your smartphone. This is what got this technology going. Some guy was sitting there going, I wonder if I can modify this app on here and run it through filter photometry and see if we can get anything that'll pick up bacteria. And there you go. This is only 5 years old or so, okay? And this helps us determine how we're gonna debride wounds, right, in the laboratory. So traditionally, we'd take this, we'd swab it, we'd culture it, we'd wait for the culture to grow, and then we can determine what kind of prescriptions we give. That takes a long time, all right? That was the analytical. So now we're talking about post-analytical. And we've all heard the stories, right? Companies having an app, people use this app, but it's not necessarily profitable to the company, right? They don't know exactly how they're gonna use this. But they got a lot of information though. They got a lot of data. And so what they do is they store that data, and someone like Google will come along, right, their Google Analytics team, and say, we don't know what we can glean from this, but we'll buy it from you anyways. So data banks are a huge part of healthcare now. And we code the data, so it takes all the personal information out, and then we send this to researchers, and they can take all of our lab information, right, and meta-crunch this information and hopefully find out something more important, okay? So hospitals are saving all their data and they're selling it. And the IT professionals that are associated with hospitals, right, are getting paid lots of money to help manage this data, right, and manage the actual data banks that are associated with it. And they're usually all on site for the hospitals, or one will be on site for a system, and they're secured and they can't be hacked from the outside because they're on premise. And I find this pretty interesting. The reason I find it so interesting is my IT friends are making so much more money than I was in the laboratory, and I wanted to figure out how to get into that. So if you have any propensity for IT or statistics and analytics with computer code, I would get into that. It's a very well-paying job, okay? So real quick, what does this apply to? And I want to briefly talk about the different disciplines that exist in medical laboratory sciences. And right now I'm just gonna be hitting the pillars. There's a couple other subsections that you guys might know about, but I'm not gonna talk about those too much today, okay? So clinical chemistry, we have a couple clinical chemists here working on a automated platform, and maybe someday that'll be replaced by that cool robot, but either way, we have to interpret those results. So what we do in clinical chemistry, like I said before, is we look at all the non-cellular components, right, in our blood and body fluids. These are glucose, creatinine and BUN, right? Kidney function tests, byproducts of protein metabolism. Our electrolytes, we have some blood gases on there. And all of these are really, really important. This guy's got a little high bilirubin, probably beat that liver up a little too much on a Thursday or Friday night, okay? And that's all that's the matter with this person, but usually pretty healthy, okay? So that's what we do in chemistry. Now, if you guys know something about this, this is what's called the complete metabolic panel. It's a basic metabolic panel plus a liver panel, okay? That's what creates a complete metabolic panel. There's a lot of tests on there, right? Every single person that comes to the hospital gets either this or a BMP, plus hematologies, plus urine analysis, plus maybe some micro if they have an infection. And more times than not, this is a normal patient, okay? Very rarely do you have a truly sick person that's going to show you some really odd values. Not that rare, but rare enough. And the reason I'm showing you this is now add 2 billion more people, right, to this system that we have, and we're going to have to be doing a lot more of these. And we're going to have to be doing it with less, which means the amount of blood we take out of these people, right, is going to have to go a long way. You guys ever heard of the company called Theranos? Ring a bell to anybody? Theranos was what they called a Silicon Valley darling, which means it's a company that's going to make a lot of money for a small group of people. So Theranos, their whole spiel was we can do hundreds of tests on a very small amount of blood, okay? And this was the beginning of this true nanotechnology, where we're using nanoliters instead of microliters. And Theranos was trying to get a CMP out of less than a microliter of blood, which is just a speck if you look at it with the visible eye. And Theranos didn't have the technology down. People caught wind of this, and they shut down overnight, practically, okay? So the reason I say this is, even though innovation drives this market, it makes us extremely susceptible, okay? It makes us extremely susceptible to market woes. So if the technology is not good, there will be someone that figures that out, and you will go to the wayside. All right, so hematology, or what I like to say is the study of the cellular components of our blood. And in hematology, we do lots and lots of blood differentials. We take your blood, we put on a slide, we stain that slide, and we look at it underneath a microscope. We're concerned about the shape and size of your cells, both red and white and platelets, and we spend a lot of time manually differentiating these cells. As a hematologist, you will sit in your chair for several hours on a shift, and all you're doing is have your head down in your microscope, and you have your little counter over here, and you maintain this posture for hours. Sometimes you can go all night without lifting your head up, okay? Which is nice sometimes. Sometimes you don't want to talk to people on a Monday. I'm totally fine with working on the diff bench, okay? Don't talk to me, leave me alone. So the reason why automation and robotics will be so important for this process is because, again, we rely on human beings to do the bulk of the complicated work. Machines already can count these cells, by the way, and they do a pretty darn good job of it until it gets real hardcore. And then we rely on the humans to do this because the machines just aren't good at it enough. And the one piece of technology I want to tell you about is live cell analysis, right? So flow cytometry, which utilizes a sort of a channel, this electrical channel that lines up cells, right, and is able to count them based on their relative shape and charge and the antigenic determinants they have on their surface. And we're able to do this very, very quickly. However, the technology is still not there. It's very, very slow. So that's what's going to be applied to hematology. So micro. Everyone likes micro because it's so manual. You feel like you're cooking with your Easy Bake Oven for the first time, right? You're feeling really excited about this brownie you're going to make. And that's how I feel about micro, is you get to use your hands a lot. You see the product. You see the culture, right? Everything is right in your face. Now, the problem with microbiology, though, is we have to wait on nature for these things to grow, which makes it difficult to treat patients sometimes. Makes it extremely difficult. We still practice prophylaxis, right? So if we have an idea what a patient's on, we just give them some broad spectrum antibiotics. And that's a problem, by the way, right? That's a problem. Just sort of, I get a feeling you might have an E. coli infection. I'm gonna give you something that'll wipe out everything on Earth, all right? Let's do that instead, okay? So the problem with that is that sometimes people don't finish their antibiotics and we create these superbugs, right? And it's gonna be even more hard to identify them in the future. So what do? Well, we use the most up-to-date technology that's available to us. I like to introduce to you something called the MALDI-TOF. Matrix-associated laser ionization by time of flight, right? There's a mouthful for you. I practiced that a bunch today, by the way, okay? So the MALDI-TOF. What the MALDI-TOF does is very, very interesting. This is the most up-to-date analyzer that exists right now, presently, okay? So you walk in a laboratory and they got a MALDI-TOF, people are usually pretty happy working in that laboratory. So what does it do? So generally, what we did in microbiology is you have a oozing ulcer or a wound or a canker or whatever it is that's infecting you. We swab that wound, we take that swab, and we streak it to blood, McConkey, and chocolate agar as our initial primary sub, that's what we call it. And then once those colonies grow, we look for predominant colonies on the agar. When we can isolate those individual colonies, right, you can see them sort of separating out at the end of your quad streak. You take those individual colonies and then you run those colonies under a series of biochemical tests. And based upon those results, we can determine whether or not a patient has a particular kind of species of bacteria, okay? So it helps us speciate. Now, this is a tech sitting at a bench with test tubes, taking individual colonies and doing biochemical tests on them, right? One at a time. This is a very slow process and it's reliant completely upon the human being, right? I like human beings, but they will fail you, okay? They will fail you. So this is where the MALDI-TOF comes in. We take this colony, and instead of running our own biochemical tests on it, or even putting it on an automated biochemical platform like a Vitek, we are going to blast it with a laser beam instead. That's always my option when things are not working out. Let's blast it with a laser beam, take a look at it that way. So, and literally that's what we do, right? We take the colony, we blast it with a laser beam, and that bacteria that is made up of glycoproteins and lipoproteins and phospholipids, it is broken up into pieces. And as these pieces fly through this matrix, an electrical field generator polarizes these particles, okay? Now the lighter particles with a higher charge tend to float further down the matrix and a little bit longer, right, than the other particles do. These protein fragments all have their different characteristics in the time of flight scenario. So what we do is we look at those kinds of time of flight patterns individual to certain bacteria. And instead of sitting at the bench and doing biochemical reaction after biochemical reaction, we stick it, the laser beam blasts it, and it takes maybe 15, 20 minutes to analyze it, and we speciate it. In the meantime, we use 0 consumables, by the way, right? No indol reagents, no peroxide reagents, no coag reagents, none of these things we needed to use, and this can do this one after the other. I will say it's not perfect. It has a hard time picking up on just random E. coli, okay? So the technology is not perfect, but it's here. And this is based off of mass spectroscopy, by the way. It's not anything different than a tandem mass spec, besides the MALDI portion, okay? So that's technology for micro. So, but we still have to grow the samples, right? Okay. So instead, let's just take the genetic material, right, and identify the genetic material. This is where molecular diagnostics comes into play. So traditionally in micro, with infectious disease, we have to grow the organism. Or if the organism won't grow, like syphilis or Lyme disease, I have to look at the antibodies that you created by being infected by it, right? That's what we do. We do it by indirect methods. That's immunology, by the way. Infectious serology, to be specific. So this is our workaround. We are not going to grow the specimen. The specimen is already growing inside you, right? So I don't need to grow it anymore. What I'm going to do is I'm going to look at your blood, and we're going to find that genetic material from this virus or fungus or parasite or bacteria, and we're going to look at that genetic material instead. And that's what molecular diagnostics does. We identify the nuclear material, and that will sequence itself out, and we get enough nuclear material that we can give you a definitive, yes, you have gonorrhea. Sorry about that, okay? And by the way, we do use this platform for gonorrhea and chlamydia. For a long time, because those are very hard organisms to grow, right? We've been trying to find a way to do this for a long time. So I graduated ASU in 2006, 2005, 2006. And we didn't even have a molecular diagnostics discipline in my program. This is how new this stuff is, okay? So it hasn't been around that long. It's cutting edge. and it's not replacing any jobs yet. Okay. My least favorite sample matrix, I'd rather deal with sputum or feces, but urine is the worst. I'm sorry, it really is. And I'll tell you why. Because the cups that they put them in are not leak-proof for some reason. You think that they'd get their heads around a leak-proof container for a urine matrix, but no, they leak like crazy. And then you get urine all over yourself, which is, that's not fun, yeah, not fun. So in urine analysis, that's exactly what we do. We look at your urine, right? It is very telling what you're doing in your life from looking at a person's urine, okay? And more times than not, we take the urine and we compare it to the blood. And we need the comparison between these matrices to find out if, say for example, your kidneys are functioning properly, right? Because if your kidneys are burned out, that urine is really, really dilute, okay? It's not filtering anything it's supposed to. Or if you have a hyperfunctioning kidney, right, you have a very concentrated urine specimen. So, technological advances in urinalysis. Mostly what we see is the spectrum of urinary tract infections, right, being able to be identified in a matter of 3 hours rather than waiting to grow the E. coli out on a McConkey auger, okay? So, if you go in, you have a urinary tract infection, they make you pee in a cup, we take that pee and we streak it on a plate and wait for it to grow. Now we don't have to do that at all. We put the urine matrix in the molecular diagnostics machine, it sequences the DNA for us. Again, bam, you have gonorrhea, okay? So, the last thing I would like to talk about, the last discipline in medical technology, and it's not the last discipline, but it's one of the major pillars, as we like to talk about, is transfusion medicine or blood banking. And in transfusion medicine, that's exactly what we do. We're transfusing blood from one person to the next. But it's not an autologous transfusion. This isn't your blood that you're putting back into yourself, which is something that people do to prepare for surgeries, by the way. No, we're making sure that the blood from another person will not react with you, right? Graft versus host disease, which could kill you, right? Having a transfusion reaction is not something you guys ever want to go through. Luckily for us, blood bank is built off of the premise of duplicate and triplicate safety measures, right? We're always making sure that some sort of practice is in the background so we don't confuse specimens, so we don't mess things up. We want to make sure that we're giving the right blood. Now, with blood banking, this is still a very old school manual practice. Now, we've automated some aspects of it, but I still have to take your blood, combine it with your blood, and see if they work, okay? And we have a feeling that blood bank is going to stay this way for a while. It's going to stay this way for a while until we can start engineering some O-negative blood, right, out of blood farms, and people won't have to give their blood anymore. But that's going to be in the future. That's like way in the future, okay? So, I see him mouthing blood farm. Yes, a blood farm where we actually grow blood, we package that blood up, and we send it out. I'm harvesting blood. I work at a blood farm. So, career opportunities. What do we do in the laboratory in terms of how are we going to make money? What kind of jobs can we get out there, okay? So what you'd usually do is you'd be a generalist, which means you work in hematology, you work in chemistry, you work in urine analysis. You do a multitude of things, and you, generally speaking, can do pretty much everything, with exception to blood bank and micro, okay? Those areas are their own specialty areas. You have to spend some time in those areas before you get competent enough to work there on a consistent basis, all right? So you start out as a generalist, and then usually you can specialize into some other area. 2/3 of us work in hospital labs, okay? So the majority of us work in hospitals. The rest of us work in veterinary clinics, they work in research labs, clinical trials laboratories, which is still considered research. And then there's everything else out there. Commercial labs, labs that do testing on topical stuff, labs that do testing on makeups, right, and sunscreens. All these things need lab technologists to test them. And if you folks want to join the Peace Corps for a year, more power to you. That's a volunteer thing, so you're not going to be paying off any of your student debts while you're in the Peace Corps. Another thing we can do is we can help provide ancillary services to our lab techs, right? By one, going into sales, which is extremely lucrative, and then also servicing these big platforms that we have, the automated lines, the robots that are soon going to be replacing the techs. Not really. But what's nice is with service is you're constantly tinkering. So if you're a person who likes to work on your motorcycle, right, or anything like that, and likes to tinker, working on these analyzers, they're pretty interesting when you open them up. I'll be honest with you. I grew up, before going to college, I worked in motorcycle shops. That's what I did. I worked on bikes. And I learned more from working on an old two-stroke carburetor, right, in terms of working on the analyzers than I ever did in school. So those of you who have an extreme mechanical aptitude, this stuff is for you, most definitely. Okay, you can go the big boss route and become an administrator in your laboratory and lord over all your lab techs and make sure that they're doing well or make their lives a living heck, one or the other. You can be the overlords of the overlords and be a hospital administrator. Some people from the laboratory will get into infection control, which is an interesting job. And then education, like myself. I like doing this. I feel like I have the heart of a teacher. I like training people. I like getting people jazzed up about what I'm doing on the bench. Okay? And if you have that passion yourself, if you're excited about what you're doing, I suggest you pass that on to somebody else. Okay? Essentially, we got to keep the fire burning, right, as time goes on. And then I threw this on here because I got to do something like this. Using my skills as a laboratorian and understanding quality processes, I convinced the largest hydroponic tomato grower on the West Coast to hire me as their botanist. Okay? And I did that for 3 years. I grow the best friggin' tomatoes you've ever had in your life. And it's all because I understand systems, right? That's it. I knew nothing about botany, knew nothing about the stomatas on root fibers, but I learned all this stuff because I knew that I would be able to learn it, and because of my experience in the laboratory, I can see the parallels that exist everywhere else in the world, okay? So you start to see things in a little different fashion. You start to see things in both a human, empathetic way, but you also see data, too, right? And that's what appeals to me. I love data. I find patterns in the most random things, and that's interesting to me, okay? Alrighty. And just to give you an idea of what the pay scale is in laboratory. So a bachelor's degree in medical laboratory sciences will start you out, on average in the United States, about 63 grand a year, okay? Not bad. About 30 bucks an hour. And in Oregon, it's even more, okay? So we like to take care of our own here, especially in healthcare. Now, if you want to continue to move up, right, you can become a manager or a chief technologist, $70,000 to $77,000. The LIS manager, right, when I was telling you about the people who are involved with IT in the laboratory and the data storage, $83,000. By the way, an LIS manager at the VA is a GS-13 rank, and they make $112,000 a year. And it's the most kickback, chill job you've ever had. So if you guys decide to go into this, check that out. And then lab directors can make over 6 figures, okay? And the evolution from a bench tech to a director, if you play your cards right, is only about a decade. And you're continually improving yourself, right? Your education, your knowledge base, and you find yourself moving up. You have to try to remain complacent in the laboratory to keep your current level of job without moving up, right? The next generation that comes in requires you to take a position a little bit higher. So for the most part, it is, I hate using this term, organic, that you move up in the laboratory, okay? And that's it for me. I'm going to pass this off to Deb. She's going to tell you a little bit more about the program. I appreciate you coming in today, okay? If you guys have any questions, thank you.